Next thing that we're going to be taking a look at here, folks, is the first part of learning target 3.2. This is the second learning target in this unit, and this one really focuses on DNA replication, or taking one strand of DNA and using it to create two identical strands of DNA. This is going to be happening on page 6 of your learning targets, and we're just going to cover subtask 1 right now. Now, the big idea here is that when you are copying your DNA, there's a process, and there's going to be a lot of enzymes, and each of them is going to have one small, very specific job. But if I were to show you this process from beginning to end, the real idea here is that what we're going to want to do is have a strand of DNA, and this is going to reset here in just a moment, but we're going to have a strand of DNA like the one that I have in this model right here. And in order to make two identical strands of DNA from this one, there it is, you're going to split that double helix in half, basically ripping the base pairs apart at their weak hydrogen bonds. Then you use the open strand to create a new half strand on both of them. And if you unzip the entire double helix and you use each half to create a brand new half, you will have two DNA strands, and they should both be the exact same code, if no mistakes are made, if there are no mutations. Now, in this case, what we are going to be starting with is when this happens and broadly what this looks like. Cells go through a life cycle. So when a cell divides and creates two brand new cells, each cell basically starts over in its own little life cycle. Think of it as a baby cell. Then that cell grows up, it matures, and then at some point it reaches the end of its life when it's going to divide and create two cells again. So this circle right here is showing us the cell cycle. And right here, right before this little label called G1, this is the beginning. And right here, a baby cell begins to go through this cycle that it will repeat. The first phase is called growth 1, and that's G1. Cells grow, they get bigger. Then you enter S, and S stands for synthesis, or DNA synthesis. That's the part of the cell cycle where cells are going to do this process and create two strands of DNA from one. After synthesis, cells go on to growth 2, they grow some more, and after growth 2, that is when they might actually do their cell division. They might divide. So right here, P, M, A, and T are all different phases of mitosis, and at the end of that, you create two cells that start this cycle again. Now, I do want to point out over here, when you have a DNA strand and you've unzipped it, you can actually refer to the original DNA as parent DNA, or parent strands. The new strands that you create from those templates is called daughter DNA, or they are daughter strands. Now, this is going to create two strands of DNA when there used to be one. And I'll remind you that chromosomes are actually just made of two strands of DNA. One strand is right here, the other strand is right here. It's one long strand, and then they're connected to each other at a centromere. Again, these are not usually visible. The only time you're ever going to see a chromosome is when all the DNA is condensed right before mitosis. So it would be at the end of G2 when all your chromosomes start to condense and appear. So these two molecules that are created inside of the nucleus, this is where synthesis is occurring. You're going to be doing your replication inside of the nucleus of cells, and that's going to have a lot of enzymes, and you're also going to need a lot of free nucleotides, those monomers. They're floating around, they're unconnected, and you're going to have to put them all together in the right sequence. Now, complementary base pairing is crucial here. If you didn't have complementary base pairing, you would not be able to use only half the DNA to make the other half. So it's important to remember Chargaff's rule and how important it is. Now, ultimately, the enzymes of DNA replication is going to be something that requires a little bit of studying and a little bit of work. Each enzyme has a name, each enzyme is a protein, and so each protein here is going to have a different job. The first thing that we want to take a look at here is how DNA replication ultimately works. So if you were in class, I would have you do this on a whiteboard just to sort of model the process as a whole. So what we have right here are two strands of DNA, and I'm going to use a black marker here to indicate that the DNA we are talking about is the parent DNA, the original DNA. Now, the first thing that you're going to have to do is know which sides are 5s and 3s. So I'm just going to assign one side as 5 and one as 3. That means on the other side here, there's going to be a 3 and a 5. And replication has to happen from 5 to 3. The overall process here is, the first thing that's going to happen is you're going to unzip your DNA. So I'm going to erase this and I'm going to redraw the last half of this as separated strands of DNA now. They have been split apart. The hydrogen bonds are broken. This is still a 3' end here. This is still a 5' end here, which means that some of this DNA is going to be built in one direction and some is going to be built in the other. And that's what's going to lead us to this thing called a leading and a lagging strand. Right here, it just so happens that if I start to build new DNA, this DNA right here is going to have a 5' end, and the 3' end is going to point that way. On this top strand, I can start here and build this direction. On the bottom side, if I look at the direction of 5 and 3 on the new strand, it's actually going to point away, which is going to be a little bit of a problem later on. Now, all of this DNA actually needs a starting point. And so we're actually going to use a little bit, because I'm going to have to use green here, a little bit of green to indicate RNA. And RNA is going to be our starting point. So there's going to be a little bit of RNA attached to the DNA for a moment. Over here, we only need one piece. But over here, we're actually going to need multiple pieces, so I'm just going to go ahead and draw in 3 of them. Now what we can start to do is build in our new DNA. New DNA is going to be represented here with blue, and I'll call this daughter DNA, or the daughter strands. So again, over here, I start at the RNA, and I can go this direction, and there is a direction here. I'll put in a little arrow to show that direction, but that direction is, if this is 5, then that means over here, that's the 3, this is the 5' end, and that makes this the 3' end. That DNA has been built from 5 to 3. And it can just keep going as this double helix continues to unzip. Down here, over here, this is going to be the 3' end. So I'm going to start at my RNA, and I actually have to build backwards away from the unzipping. That's my 3' end, that's my 5' end, so I've built a little chunk. I'm going to have to do that over and over again. Here's another chunk. I'll start at the RNA, and I'll go this direction. Again, building from 5' towards 3'. And I can get one more chunk in here. Again, 5 to 3, going this way. And as this opens up, I can keep building new little chunks, tiny little chunks as we go. This one, I really only need one long strand of DNA. So ultimately what this does is this creates one strand that is easy to build in one long go, and one strand that's tough to build as you have to build it in little chunks as the DNA becomes unzipped and available. Now ultimately, from here, we can actually determine which strand is going to be easy to create and which strand is hard to create. Over here, if this is the 3' end, then I know the new strand is going to have a 5' end here. It can go all the way this direction. That is the easy strand to build. That strand is going to be called the leading strand. Our other strand over here, if this is 5', I know that on the other side, that new strand is going to be 3'. So if I'm building from 5 to 3, I actually have to go this way, backwards. That's going to be a little trickier, has to be done in short chunks. That's going to be called our lagging strand. Which brings me to this final idea in Subtask 1. The strand that is built continuously towards helicase and the expanding replication fork of DNA is the leading strand. The strand that must be built away from helicase, away from this fork, going the opposite direction, that is called the lagging strand. And that's going to do it for subtask 1.